What if I told you that right now, inside your body, roughly 3.8 million cells are dividing every single second? That's not sci-fi, that's biology. And today, we're cracking open the one chapter that NEET loves to test again and again: cell cycle and cell division. From G1 to G2, from prophase to telophase. By the end of this video, you will never confuse mitosis and meiosis again. Let's go. The cell cycle is the sequence of events a cell goes through from the time it's born to the time it divides. It has two major phases: interphase, which is G1, S, and G2, and M phase, which is mitosis and cytokinesis. Now here's a neat trap. Interphase is not a resting phase. The cell is extremely active, growing, duplicating DNA, and preparing to divide. The only true resting phase is G0, where cells like neurons exit the cycle permanently. Remember, G0 equals gone from the cycle. S phase is where the magic and the neat questions happen. During S phase, DNA replication takes place. The chromosome number does not change, but the DNA content doubles. So if you had 2 N chromosomes before, you still have 2 N after, but now each chromosome has 2 sister chromatids. This is a classic NEET MCQ trap. They'll give you a scenario and ask, at which stage does DNA content double? The answer is S phase, not prophase, not metaphase, S phase. Let's walk through mitosis, the equational division. Prophase, chromatin condenses into visible chromosomes. The nucleolus and nuclear envelope disappear, and the spindle apparatus forms. Metaphase, chromosomes align at the metaphase plate. NEET loves this. Metaphase is the best stage for karyotyping because chromosomes are maximally condensed and clearly visible. Anaphase, centromeres split, sister chromatids are pulled to opposite poles. Telophase, everything reverses. Nuclear envelope reforms, chromosomes decondense, nucleolus reappears. And finally, cytokinesis divides the cytoplasm. Result? 2 genetically identical daughter cells, both diploid, both perfect copies. Now for the big boss, meiosis. This is where NEET drops most of its marks in this chapter. Meiosis has 2 divisions: Meiosis I, the reductional division, and Meiosis II, which looks like mitosis. The most important stage? Prophase I. It has 5 substages: leptotene, zygotene, pachytene, diplotene, and diakinesis. Crossing over, the exchange of genetic material between homologous chromosomes, happens at pachytene. This is the source of genetic variation. NEET has asked this at least 3 times in past papers. Pachytene equals crossing over. Tattoo it on your brain. Let's lock in prophase I forever. Use this mnemonic. Let's zoom past deep darkness. L for leptotene. Chromosomes first become visible as thin threads. Z for zygotene. Homologous chromosomes come together in a process called synapsis, forming bivalents held together by the synaptonemal complex. P for pachytene. This is the big one. Crossing over happens here at points called chiasmata. D for diplotene. The synaptonemal complex dissolves, but chiasmata are still visible. And darkness, diakinesis. Chiasmata move to chromosome ends in a process called terminalization. Nuclear envelope breaks and spindle forms. 5 stages, 1 mnemonic, 0 confusion. This table is gold for NEET. Print it, photograph it, stick it on your wall. Mitosis happens in somatic cells, body cells. Meiosis happens in gonads, testes, and ovaries. Mitosis has 1 division, produces 2 identical diploid cells. Meiosis has 2 divisions, produces 4 unique haploid cells. Mitosis equals growth and repair. Meiosis equals sexual reproduction and genetic variation. If NEET gives you a scenario with genetic variation or gamete formation, the answer is meiosis, always. And that's a wrap on day 11. You've just mastered one of the highest weightage topics in NEET biology, cell cycle and cell division. Here's your 60-second checklist before you sleep tonight. G0 is the exit phase. S phase is when DNA doubles. Metaphase is the best stage to count chromosomes. Pachytene is when crossing over happens. And the 5 substages of Prophase I are leptotene, zygotene, pachytene, diplotene, diakinesis. Let's zoom past deep darkness. If this video helped you, smash that like button, subscribe, and drop a comment with your score on today's practice questions. I'll see you tomorrow for day 12, the big revision day. You've got this.